Uh, uh, Pointed gourd is majorly cultivated in Indo-Gangetic plain, and it is majorly cultivated in other eastern parts of India, and especially in West Bengal. And this economically important crop is cultivated around the year, but especially mostly the cultivation happens during the May to October, and which coincides with the rainy season. Many biotic and abiotic stress affect the yield of pointed gourd, but the major constant is Phytophthora melonis, which is the less studied pathogen and Oomycetes pathogen. So, this Phytophthora melonis causes actually the fruit and vine rot disease of pointed gourd. And the major yield limiting factor of Phytophthora melonis causes severe crop damage up to 90%. It was reported that the center of origin of pointed gourd is in the Bengal and Assam area of Indian subcontinent. And Phytophthora melonis was reported from different parts of Asia and from different hosts like cucumber, pistachio, and 4 other different hosts, and also from pointed gourd. And this is for the first time in India, which was reported by our lab in 2006. And it was stated that the geographical origin of Phytophthora melonis is Southeast Asian countries. So it is expected that the Phytophthora melonis may be originated from this region, and the diversity of this population in India would be more diverse. So. So we all know that to get the control strategy are affected by the diversity of the population. So to do that, a robust control strategy is needed, which target the population instead of the individual. And to understand the phenotypic and genotypic variation among this population, we need to have the population genetic structures of Phytophthora melonis. So we all know that to do the any kind of population study, we need to have the robust and extensive sample collection, and we did the sample collection from the majorly pointed gourd cultivated states from India, and which is West Bengal, Bihar, Jharkhand, Uttar Pradesh, Odisha, and Assam. And we have collected more than 150 samples from these different parts of India. To approaches for the population genetic study, we need to have the proper genetic resources in our hand. But in our case, there was no genetic resources available of Phytophthora melonis from pointed gourd in India. So we first did the whole genome sequence, and we have assembled the sequence, and the sequencing was done on the isolate collected from Hooghly. And the assembly report was submitted to NCBI, and the isolate deposited to NCCS Pune. And what we got from the assembly, the genome assembly size of our isolate was nearly about 63 MB, with the phospho completeness score was 100%. But in other Phytophthora melonis isolates collected from the other parts and other hosts, they are showing the phospho completeness scores less than 100%. And one interesting fact is that our isolate is showing 53% of the repeat sequence of total genome, and this makes this isolate the third largest Phytophthora in terms of repeat sequences among all Phytophthora. So, and this is the first sequence of Phytophthora melonis from India, from pointed gourd. Then after getting the assembled sequence, we did the in silico analysis of whole genome screening for SSR motifs, and we got nearly about a huge number of primers, that is nearly about 5,000 primers, and we screened the 5,000 primers on the sequenced whole genome. By EPCR analysis, we got some primers, and we narrowed down the number of primers. After getting these primers, we did the wet lab validation, and we finally selected 8 primers from the population analysis of the whole population, and we selected 8 number of primers, distinguish the whole population. And further, we have checked the fungicide sensitivity assay, because still yet the fungicides are recommended for to control the disease. So we selected 5 different frag group fungicides from different single gene targeted fungicides, and we have taken azoxystrobin, mendiprobamid, etavoxam, metaloxyl, and cyclopamide. And out of these five fungicides, etavoxam and cyclopamide are 2 newly introduced fungicides in India. And what we got from this in vitro study from the population screening, we got the population is highly insensitive to azoxystrobin, and the whole population is highly sensitive to mendiprobamid and etavoxam. But in case of metaloxyl and cyclopamide, we got some sort of variations in responses. So what we can recommend, that is based on this in vitro study, but this is a very preliminary study, that this azoxystrobin cannot be recommended for farmers use, and mendiprobamid and etavoxam can be used, and but we can say it after the field trial. So lastly, I would like to thank my supervisor, Professor Sanjoy Kuharay, Department of Botany, and Dr. Sujita Tripathi, IICB Kolkata, and my lab mates, Dr. Tanmoy De, Mr. Soumodip Ghosh, and CSIR for my fellowship grant. Thank you.